So the last phylum in the Cebus vascular plants is the Polypodiophyta, which is, I think, how your book is expressing that now, but there are other names. You might see Terophyta or Pteridophyta. And so now this is probably the group you're most familiar with in just living your ordinary lives. You've probably seen a fair amount of different ferns. And when you add them all up in terms of the number of species of ferns, there's actually more of them than there are gymnosperms. Most people think angiosperms or gymnosperms are the big winners in terms of just sheer population sizes and number of species. But the ferns are quite diverse, and they are able to, a large part of that is due to some of their rather interesting plant genetics that they have, and which I won't get into, but they have some very interesting mechanisms and extra chromosomes to do interesting things with. So of all the Cebus vascular plants, they are certainly the most diverse in terms of where they can be found, as well as their body design. Their body design can be quite diverse, and so their morphology. If you really want to get to know the ferns, go from here. You got to go into the tropics. That's where 3/4 of the species are found. And then when you're down there, look up, because you have to look up into the forest canopy, look at the branches of trees, because that's where a lot of the epiphytes, ferns are existing as epiphytes. There's been some interesting forays into tree canopy research, which I think would be so fun to try. I've seen a few videos and heard one person give a talk on her graduate work on essentially creating this scaffolding system very safely for her as well as for the trees and plants. She was studying the ecosystem of epiphytes in a tropical rainforest, and it was just fascinating. Of course, lots and lots of ferns and mosses and other bryophytes. So lots of interesting plants existing on trees and just, living their life. They're not harming the tree. So you can have, in terms of diversity and size, you can have ferns that exhibit leaves that are 9 meters long by 4.5 meters wide. I once saw a picture, which I can't get my hands on. It's somewhere on the vast internet. There was an old-fashioned, one of the older models of a VW, a Volkswagen Bug car, with this leaf draped over the top of it from the very top, front of the car to the very back of the car. I can't get my hands on that photo somewhere in Google land, but amazing how big that leaf was. And then you go down into a few millimeters for some of these other ferns. And so Salvinia and Azolla are two aquatic ferns that have very delicate, petite looking leaves. And so here we tend to have a lot of bracken fern that really does dominate a lot of our areas here. And so Tritium species. And so many of the ferns, when you look at them around here, they grow through rhizomes. So they have underground stems, the rhizomes, that will pop up new fern fronds every year. And so it's just asexual regrowing of the fern frond. And so if you're trying to get rid of your bracken fern in your plot of land, you're going to be in for some hard work, because you have to dig up the rhizome. It's a lot of work. So this is a little challenging. Like I said, I wish I had the VW Volkswagen Bug picture, but this is the giant fern frond Boracia. And then you have these giant tree ferns, not around here, unfortunately. You have to go south for this, or Australia or New Zealand. But tree-like fern species, just beautiful, beautiful species. Here's another image. I think this one is New Zealand. And so they are all over the world, from big to small. Very interesting leaf design, typically. And yes. Awesome. Mm-hmm. Mm-hmm. Yeah.
Their issue when how does that transition happen? Why does it get lignified?
I mean, they do have Xylem. They have the trachid version of Xylem. So, they can get heavily lignified. As far as stem tissue for tree ferns, I'd have to look closer at that. I'm not an expert with tree ferns in terms of their anatomy. Like, I'm pretty sure there's some big differences compared to like, I don't think no, I don't think so. And that's another thing I would have to confirm. I could ask Dr. Rotter. He teaches plant anatomy at his current institution. So I was actually asking him just last hour. I met with him. I'm like, so you teach plant anatomy? I was like, that'd be great if you could do that here. So anyways, we used to have a plant anatomy class for a whole semester, just plant anatomy. And then when we had people retire out, we weren't allowed to replace that person. So we don't teach it anymore. So it'd be nice to have that. But, I think with tree ferns, they certainly have enough growth over the years with their xylem to get that transition. But again, I'd have to look more carefully. And I'll do that. I can look into that, but give you some feedback. So where did you go over spring break? Oh, Puerto Rico. Tree ferns. Awesome. Yeah. So um these are aquatic ferns. This is a patch of Azolla. And so each leaf you can see is very petite. That's a big whole cluster. So these are many, 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 many tiny leaves all in this image. Okay. And I'll talk more about Azolla because they're very cute, but they can be troublesome in different ecosystems. Salvinia is on the left. That's another aquatic fern. Its leaves are a little bit bigger than Azolla, but again, that's a big patch of many leaves of Azolla floating. And then there's some beautiful staghorn ferns that you can see in different environments. We used to have one in the greenhouse that was just gorgeous. Lately, but so lots of different morphologies in terms of leaf design. And basically, with this picture, really, I'm just trying to get you to think about the fern frond. And that's a compound leaf. I don't really expect you to know the jargon on here, but that becomes important if you're going to do any identification in the field. So if you have a field guide with you on plants, they will ask, they'll use this jargon and ask you to look for this type of thing. But fern fronds are definitely compound leaf. Okay, so when you're out hiking around and you see those fern fronds, that's the sporophyte that you're looking at. And like I said, every spring, it's a great pleasure to see them growing, poking through the snow. Usually, well, we don't really have much snow, but usually around here, there's traces of snow left on the ground, and you'll see these fern fronds popping up from underground rhizomes. And they are in a different shape. They're in a fiddlehead shape, which is essentially a very coiled fern frond that looks like the end of a violin, the fiddle. And so they they think that they are assuming that very coiled growth form so that they can push through the leaf litter, any snow, and they can easily push through that without damaging their fern fronds. And so what happens is they'll pop above ground in that very coiled up shape, and then they have to undo that once they get above ground. And so it's this asymmetrical growth pattern that has to kick in to kind of uncoil and unfurl so that they can get their fern frond fully out and ready to go for photosynthesis. And so when they are growing underground, they are coiling up by asymmetrical growth, and when they pop above, they got to reverse that and uncoil. And so when you are out hiking around, you may see some before the end of the winter semester. You may see some fiddleheads growing up out of the soil before you finish your semester. Most fern fronds do die off though every fall. But again, the rhizomes are there, and next spring they'll pop up again. They don't have strobili. So, when we were looking at the club mosses and the ground pines, they usually very easy to see their strobili. The horsetail ones, you can see their strobili. Fern fronds don't have strobili, but they still do spores. Okay, they're still seedless. They still form spores. So we'll talk about that. So here are some fiddleheads. They're so cute. You can see them popping up in the undergrowth when you're hiking around. And so you can see how this is, much more better shape for pushing through some soil, leaf litter, potentially some snow pack. And then once they get up, they go on. So some people will forage for these and harvest them to eat them. And there's been a long history of that. I've never done it. Has anyone done that? And I when people who have done that, they tell me they usually like fry them up in garlic and butter. Is that kind of the standard? You have to boil them first. Okay, boil. Saute them. Okay. I guess they're tasty. Oh yeah, okay, that makes sense. I have not done that myself. There was a paper published quite a while ago, and I would need to do some digging to see what happened from that, if there's been any supporting data since then. That overeating on that, there was some correlation between eating those and stomach cancer. But that was a paper that was published decades ago, and so I don't know if there's been any more research on that. But they are considered a delicacy among many people and are eaten widely. This is just a beautiful picture showing the xylem, big xylem in there under this is like the central stalk of the fern frond. Okay, so they don't have strobili, but they do have to do sporangia with spores. And so their version, instead of strobili, is sori. Talk about one, it's called a sori. I usually say things in the plural for this kind of thing. So sori, these are clusters or collections of sporangia. They're close together, but they're not as ornate as a strobulus. So most of the time, all the spores look the same. They're homospores, but the fern group can have a mix. Some species, they form homospore spores, some are heterosporous. And that's actually one way that they use to classify these different ferns and look at their phylogeny. So you have to go to the underside of the fern frond to see these. So they're not obvious unless you deliberately flip the leaf over and look. And I remember when I was a young girl, I was feeling so sorry for the ferns in my when I was out hiking, because I saw all these brown, fluffy, powdery things on the underside, and I thought it was getting attacked by a fungus. And I felt really bad. And then I learned much later that those were actually a good thing. Those were spores. So sometimes they're easy to spot. When you flip the leaf, you'll see when they're good to go, they'll look like brown, dusty, powdery things on the underside. But sometimes they're camouflaged. They're covered with a piece of tissue that's still green, and the source is underneath that. So you have to look carefully. And they don't always produce them all the time. It's only certain phases through the growing season that you'll see those. And so the fancy word for that flap of tissue that's covering up the sori is called indusium. And I'll show you some pictures of those. So they actually look at the sori underneath the fern frond, and they use that as a way to classify and look at phylogeny. So where they form the sori on the leaf does seem to matter in terms of being able to identify them and look at how related they are to each other, the different species. So they, well, here are several different fern fronds or sections of fern fronds, and they're trying to show some options here of how they form their sori and which part of the tissue. And so, you can see, and then some of these are like this one here. They're trying to show that it's covered up by some indusium tissue, and then as it matures, that indusium tissue kind of splits, and you'll start to see more of the spores underneath, the sporangium underneath. But you can see there's some obvious differences here, the way the artist drew these sori. And so that's on purpose, because we know that the design and the placement is different for different species. And that's a good way to classify. So if you take a field guide there and you're trying to figure out what these ferns are, they will ask you to do this. So what are you looking at? Those things on the underside of the leaves are a collection of sporangia. So you're looking at an intact sporangia on the upper left. And so this design seems pretty universal amongst ferns. When they do these sporangium, they'll tend to do this where they have like 3/4 of the sporangium is nicely reinforced. And then you have a weaker section of sporangium that is not reinforced. And that's done on, that's a good design, because what happens is as that sporangium starts to dehydrate and dry, it causes the weaker section, it puts a tension force on there, and it causes that weaker section to split. And then that helps swing the spores from this that snapping action as it splits open quite dramatically allows for the spores to get kind of catapulted out of there. You don't need to know the leptosporangiate. They look at, I won't go into detail on that. That's just a way to further classify. So these sporangium, the design's quite nice for spore release. And now, if you actually look at a fern frond under the microscope, this is what you would see. So this is the fern leaf tissue, okay? And then if you look on the underside of the leaf, all of this whole thing is a sorus, that whole thing, okay? That whole thing is a sorus. And a sorus is a collection of sporangium, all right? And so all of these things in there, all of those are sporangium tissue. Okay, so this entire thing is a sorus. A sorus is a collection of sporangium. Okay, and some of them you can even see the wall design of the sporangium tissue is beefed up quite nicely, and then the weaker section is much thinner. Okay. So if you did look at the underside of some leaves, you might see something like this. So again, you can see this is indusium tissue that's covering over. It's protecting the spores as they're developing. And then you tend to see the indusium tissue start to disintegrate as the spores get close to being ready to get released. And some of these, you can actually see some of the browner areas. So you can see the indusium is kind of fading away and the spores, sporangium tissue is becoming more visible. Here's a beautiful image of that entire thing. You'd say, oh, look at that sorus. And it's a cluster of sporangium. Beautiful image there, taken for a competition. They won 11th place for that one. Very beautiful image. Okay, so if these spores, these are very nice, we have nice reinforced, 3/4 of the way around the sporangium, nice reinforced, and then the weaker area. So if you can imagine that opening up, flinging open, and catapulting out the spores, then that spore hopefully is going to land somewhere and develop into the gametophyte. Okay. And so then the gametophyte is often heart-shaped, actually. If you look at it carefully, you typically need a hand lens for this. Then by the naked eye, it's kind of hard, but it often looks heart-shaped. I'll show you a picture. And they usually attach to their substrate using very delicate looking rhizoids that they hold on to the substrate. And they're usually bisexual or monoecious. So one house, that means you have both sexes on one house. I'll show you a picture. And then it usually isn't very long lived. After you get So remember, the gametophyte's job is to make gametes, sperm and egg. And once that egg gets fertilized, most of the body of the gametophyte will die. And then that fertilized egg becomes the zygote, and then you're back into the sporophyte phase. So here is a drawing of a sperm, sperm made it here to the egg. So that egg was right around here. Gametophyte. And usually the egg is close to the front of the heart, and then the sperm is further down at the base. So the archegonium and antheridium are located in two different locations. And then once you get the egg fertilized, now they're showing the sporophyte growing out. Okay, so that's the sporophyte growing out from where the egg got fertilized. So the idea of the fern alternation of, its life cycle, it's, basically the same overall concept where you have sporophyte dominating, just that we're not really talking about a stobulus, then sori, but they still have to go ahead and make spores held within the sporangium, and those spores have to go through meiosis in order to shift from sporophyte to the gametophyte phase. And then you see the spores get released, and if everything goes okay, they'll germinate into that gametophyte. And they're trying to show the archegonia and the cleft of the heart, the antheridia are a little further down. And then the job of the gametophyte, once they do the egg and sperm, they got to get the fertilization to occur. Get that egg fused with the sperm, and then you go back up into the sporophyte. And then don't forget, with the rhizomes, you have usually every spring, you'll have sporophytes just asexually growing up the rhizome. So if you are out hiking and you do see some spores that look like they're ready to grow, they're pretty easy to actually get to germinate and grow. You have to have a little patience, but we've had really good success taking fern spores from our plants in the greenhouse and getting more ferns up and running just by getting the spores put onto some nice moist soil. Here's another from a different book, again, just for study purposes. Of these images, and if that helps, go ahead and use that. But it's saying basically the same thing as what we just looked at. Gametophytes are tiny. Like I said, you typically need a hand lens. Very delicate. You see lots and lots of tan rhizoids there. They really overproduce there. And if you look at some of the really small ones under the microscope, you can see why they say it's heart-shaped. And so that dark patch there, that's the archegonia, I should say, plural. Antheridia are more in this section here with all the sperm. Here's another one. This one, it looks like most of what you're looking at here, all of this is all antheridia. Doesn't look like the archegonia are there yet. But here, there was an egg in an archegonia that must have gotten fertilized because there's a sporophyte coming out. So again, a lot of this you would need a hand lens and look, and out in nature it'd be super hard to find, but if you grow those in a lab, you would see it. If you look here, they're just trying to show the difference here. Most gametophytes, most fern species are tiny, really, really small. And then you can have, pretty massive sporophytes of the same species. So lots of beautiful ferns out there. They grow in a lot of areas that other plants have a tough time in because of the challenging conditions of that environment. They form associations with mycorrhizal fungi. They can certainly do that. And they can form associations with ants. There's a lot of tropical ferns. This upper image here is looking at ants growing in the fern tissue. And the ants will actually form their nests inside the fern. So here is this middle picture. That's like all plant tissue, but that's like the ant nest in there. So it's a mutualistic association where the ant, the feces from the ant provides nutrition, fertilizer. The dead bodies of the ant provides nutrition. The ants will spread around the spores just by, it'll get on their bodies, and then the ants are busy foraging, so the spores get dispersed. And then the ants do eat some of the spores for nutrition, the fern spores, but it's enough of a trade-off, a win-win trade-off, where it doesn't, it's a balanced mutualistic association. So even though the fern plant is giving up some of its spores for the nutrition of the ant is returning the favor and helping the fern plant disperse and protecting it. So there's some really interesting plant animal interactions when you look at the fern lifestyle in their different habitats. And then this last image on the bottom is just you're looking at chromosomes here, and this idea that the genetics of fern plants can get really complicated. Plants in general have just wacky genetics. And we say that ferns in particular have high levels of polyploidy, which just means they have more than 2 complete sets of chromosomes. So they're just really weird. And for lack of a better descriptor, their genetics is really weird. And it primarily, what they see as a result is that they will form very interesting chemicals, different secondary metabolites. So they have the ability to form very interesting chemicals that, humans are interested in figuring out what they are, what kind of uses could we get from those interesting chemicals. And so from the plant perspective, some of these very interesting secondary metabolites often makes them very successful in being able to defend themselves against insects primarily, but also some mammals that want to chew on them. And so some of these chemicals can be deadly, and some of them can be mildly toxic. It runs a whole range of nastiness in terms of how deadly these chemicals could be. So herbivores have to be cautious when they're trying to eat ferns, because sometimes they can be really packing a punch and make it very difficult for anything trying to eat them. For example, here they're showing a cattle eating a bracken fern. And this has been a problem out west with cattle ranchers that raise cattle for food. If their cattle eat too much of that, and the problem is that, they keep popping up through rhizomes. And so it's really hard. You might not really notice them until they start growing up in the rhizome, and then the cattle start chewing on them. And if you've been out west, there's acres and acres and acres of land that don't always get patrolled by the human. And so it can be a problem that the cattle can eat it without the rancher knowing about it. So it's been a pretty chronic problem. They're trying to control it with using a fungus that is specific to certain species of bracken ferns to try to kill off the fern. So that's had some success in some areas out west. This lower image is a river, believe it or not, but it's totally covered by Salvinia. So this is Salvinia molesta. This is the common name is caribou weed. And here you can see it's pretty much choking the whole river. It's just a carpet of this aquatic floating fern. And so this has been a problem in many different river systems and lake systems. And again, in this case, they're trying to tackle this issue with a particular beetle that only feeds on this species of fern and using that as biocontrol. Obviously, you don't want to dump a bunch of herbicide into these systems. So using some more friendly alternatives is what they're going for. So there's these types of, you know, basically in this case, the ferns are considered weeds because they're growing in places you don't want them to grow. And they're problematic because spraying a bunch of nasty chemicals in these areas really isn't the best solution or any solution. On the other hand, ferns have been in, we've interacted with ferns for a long time. And so, they're beautiful. The Boston fern plant on that lower left image there, that's commonly sold as house plant and is a wonderful, wonderful plant for actually cleaning up your air quality in your home. So if you have Boston ferns in your home, they're one of the best plants at cleaning up organic chemicals, volatile organic chemicals that are in your home that you don't really want to be inhaling on a regular basis. And so landscaping too, lots of ferns we use in landscaping. Azolla can be used for good things in the sense that this is the upper image of the rice paddy, and they actually use Azolla in rotation to rice in rice paddies. That's a very common way to grow rice. They'll take turns. They'll put in Azolla in there and make sure it gets a nice healthy population of Azolla. And the reason they're doing that is that they know Azolla is carrying with it a cyanobacteria and Anabina. And so it's really, they're going for the Anabina, but Anabina loves to hang out with Azolla, and Azolla is aquatic, so it does really well in rice paddies. So they go ahead and get Azolla up and running in there. They know Anabina is there. Anabina's busy fixing nitrogen. So as Azolla is hanging out in the rice paddy, you get all this lovely nitrogen fixation going on. And so it's a really easy way to pump in essentially lots of nitrogen into the rice paddy in a very safe way. And then what they do is they go in and kind of churn over the Azolla, and then they plant their rice, and the rice have this lovely source of nitrogen for the growing season. So it works out really well. This middle image here of a concrete fern, and you see the green growth over it, that is Azolla. So some people have tried this because if you have standing water, that's right for the mosquito to grow. It likes to complete its life cycle partly in the water there. And so the mosquito larvae, if they get this carpet of Azolla on the top of these standing water, the larvae won't survive that. And so it's a pretty cheap way to try to control mosquito completing their life cycle. We get some interesting alkaloids or secondary metabolites from fern plants. I mentioned the fiddleheads as a delicacy, but just be aware you might want to do a little bit of research on cancer association. It was a correlation study. They did not have strong enough data to say anything other than it's correlated, but it's something you should be aware of. And then that Boston fern idea, the air filter idea of cleaning up your air quality, NASA actually did some experiments with a variety of different plants. They did a lot of work with Boston ferns originally, and they did work where they picked formaldehyde as the nasty volatile to see, how do these plants do with cleaning up formaldehyde? And they tried a lot of different species, and the Boston fern was one of the top winners. It was so good at removing formaldehyde from the air. And so here they're saying in this study, it was a 3 meter by 3 meter area, and it was removing 1,800 micrograms of formaldehyde per hour in that space. And so when the Weston Hall was first built, although you can't open any windows in that building, according to the fire laws. They don't want you opening windows if there's a fire going on in the building, because it will just spread the fire more. So the new code is no, you can't open your windows in these kinds of buildings. And I knew with a new building, you have new carpet, new everything, and it's all off gas. When you smell that new car smell in a new car, those are volatile organic carbons that you're taking in. And some of those aren't so good for you. So I went to the local flower shop and bought a dozen Boston ferns and put them in my room, my office. And if some of you have been in my office, it's tiny. And so you walk in there, it looked like a jungle in there. But it was funny. Anyone who walked in my office, they went, "It smells so good in here." And I didn't use, I wasn't diffusing essential oils or anything like that. It was just the Boston ferns hanging out in my room doing their job of cleaning up the air quality. That was really fun. I loved that feel. It was a very tropical vibe. The one downside to Boston ferns you need to be aware of, especially living in this area of the country, they love humidity. We don't have great humidity here in the winter. And if they don't get enough humidity, their little leaflets start to rain down and fall off the plant. And then the maintenance people got a little mad at me with all the bits and pieces of dead fern leaves all over the place. So I had to donate them up, go with different. But they're great to have in your home, but keep the humidity up. Have some humidifiers going, and then they'll be happy. And they'll do some great air quality cleanup for you. Okay, so with this group of plants, back in the day, most of the plants that we see now currently that are seedless vascular plants, a lot of them were very large, very, very large, to the point where they use the word giant, the giant club mosses, giant horsetails. We still see some tree ferns around here, so they've persisted. They've been successful enough where they can persist. But most of the seedless vascular plants that we see today, and the ferns are a noted exception, they've greatly diminished in size. There's still some ferns that are pretty impressive, but the horsetails we see now are about knee high. That's as big as they're going to get. Club mosses are very petite. But the age of coal. So these giant seedless vascular plants were very successful during the Carboniferous period. And that is, the left image is Equisetites, which, that looks like a giant horsetail. Really, the current horsetails that we have look so much like that. They're just much smaller, of course. But these are what we're tapping into for coal. So when we are harvesting coal, it's these plants that have fossilized and turned into coal, most of what we're harvesting. And so it's the seedless vascular plants. So some students in the past have asked, they're like, well, what is coal? So if you're interested, I'm not going to ask about this on the exam, but if you're wondering how all those plants turned into coal, this is a little blurb on that. So if you want to read that, go ahead. But certainly we have them to thank for all of our coal production access. As far as further uses, the rhizomes of primarily ferns are used to grow orchids historically. So back in the day, when they didn't understand how to work with orchid seeds, because orchid seeds are so dust-like, you can't just put them in soil and water them. They won't do anything unless they have a particular mycorrhizal fungus with it. And someone stumbled on the idea that if you sprinkle orchid seeds on fern rhizomes, they often sprout. They didn't realize it was mycorrhizal fungus hanging out with the rhizome of fern that left the orchid germinate. So that connection took a long time to figure out. But so it's a long history of doing that, taking fern rhizomes and sprinkling the orchid dust-like seeds on that. You can still, historically as well as currently, if you're out hiking around and tenting and camping and you burn your meals, you can use horsetails if you want to scour and clean up your dishware. They used to stop furniture with fiddleheads. That was a classic use of them back in the day. You can eat them. There are uses in botanical uses, different extracts from primarily ferns for diarrhea, fever, different wounds, coughs. The licorice fern in particular is still used. I see that tea. You can buy that sore throat tea. And if you look on the ingredient list, it's actually extract of licorice fern. And it works. It's amazing. how that tea works. I drink a cup of that with a sore throat. It makes it feel so much better. But we still are, taking advantage of some of the medicinal uses. Beautiful red dyes, that red fern there, you can see, you can get some really beautiful natural pigments extracted. And then, of course, basket weaving is still a thing and very popular and can create very beautiful, useful artwork in baskets. So lots of options here to think about in terms of, and then in your book, there's a section on economic uses of seedless vascular plants, economic uses of bryophytes. Make sure you're reading those sections. I'd like you to think about that. I'd like you to be aware and able to talk about and answer questions on how do we use these plants. I think that's an important thing to appreciate. This last slide is a table summarizing the different phyla of the seedless vascular plants. And I've highlighted in yellow the current, I think these are the ones that are showing up in your book. That's the terminology your book is using. And so those are the 4 phylum names I'd like you to know and who belongs into each phylum. As far as all the columns, looking at leaves or this differentiated into root, stems, and leaves is a good column. And then type of leaves is a good one, and type of steel. The other stuff, I'm not really going to ask about. Okay. So with the leaves, of course, you have 2 options. It's either a megaphyll or a microphyll, or, well, I guess 3 options. Neither. Venations might be a 3rd option. But please start committing to memory the different phylum names for this group too, as well as the bryophytes we already covered, and then who belongs into each plant, because that will be questions, multiple choice questions as well on the exam. Okay. So the next topic is the gymnosperms already. We're going to get into those, but we'll save that for Monday. All right. So, but that's the next group of plants. Yes. Which slide? Oh, they're not in there? Oh, I'm sorry. Yes, I will do that. I will do that.